The Galien Foundation fosters, recognizes, and rewards excellence in scientific innovation to improve the human condition.
André Malraux said once in one of his novels, "Une vie humaine ne vaut rien, mais rien ne vaut une vie humaine." Human life is worth nothing, but nothing is worth a human life. All these men and women are trying to save human lives. Is there anything more noble than that?
The Galien Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year Best Pharmaceutical Product, Best Biotechnology Product, Best Product for Orphan Rare Diseases, Best Medical Technology, Best Digital Health Solution, Best Incubator Accelerator Equity, Best Startup. Around the globe, the Prix Galien is considered as the equivalent of the Nobel Prize for the industry, mobilizing an unrivaled network of Nobel laureates and top biomedical scientists. The Galien Foundation manages an independent, cross-functional, and geographically diverse program of events and sponsorship to brand the best of the best in new medicines and diagnostics. The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Roy Vagelos Pro Bono Humanum Award for Global Health Equity is bestowed to an individual, a company, an academic institution, or a non-governmental agency that has helped to improve the human condition through the application of biopharmaceutical science to problems of developing or underserved populations worldwide.
This is the right event, on the right issue, at the right time.
Staging this event in Africa is a significant statement that science and innovation are needed as much here in Africa as anywhere else.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galien Foundation.
Welcome, everyone, to this week of biomedical innovation webinars that are being hosted out of New York. Our objective this week is to provide some additional content and background for the largest annual event, the Prix Galien US Awards, which will be held this Thursday in New York. In conjunction, this will have the annual Galien Forum of Speakers representing the top scientific experts and leaders in public health, which is what we're doing here today. Because of the mission of Galien is to improve the human condition, we've developed this webinar series to give our nominees for Best Pharmaceutical, Best Biotech, Best MedTech, Best Rare and Orphan Disease, Best Digital Solution, Best Accelerator and Incubator, and Startup categories, greater visibility in describing their journey of medical discovery. This year's Galien USA Awards has been a record-setting year with 176 nominees in 7 categories across 169 different companies, covering 18 diverse therapeutic areas ranging from cancer, cardiology, and neuroscience to infectious and very rare genetic disorders, diseases, and vaccines. We're going to be looking closely at the products in all of these critical areas throughout the week. But today, we're here to talk about the best pharmaceutical category. So Our focus will be holding 5 webinars due to the large number of interest and folks that we're speaking to today of different medicines representing these important fields. This specific webinar is going to be devoted to our lung, gland, soft tissue, blood, and skin cancers, and we're delighted to have the panelists that we do today. Our task is to assess the current state of scientific progress in fighting these multi-symptomatic, complex, and hard-to-treat cases. We'll also gain some of the perspective of the nominees' companies and their experience in moving into these promising compounds as it relates to clinical trials, regulatory approval, and their market acceptance. Now, we do have some time for questions in here. There's a little Q&A piece down at the bottom. Feel free to be using that. But I'd like to introduce our esteemed panelists today. So first, we have Patrice Lee, who is Vice President at Pfizer, representing Braftovi. And I apologize if I've butchered any of those pronunciations. I probably got Pfizer right, though. We have Josh Baumel here as well, who's global medical head at Janssen for Rybrevant, and Susan Pandya from Servier for Tibsovo. I'd like to give our panelists the opportunity to now introduce themselves. Susan, would you mind kicking us off?
Yeah, absolutely. I'm so happy to be here. Nice to meet you, Robert, and grateful to be on this esteemed panel with Josh and Patrice. My name is Susan Pandya. I oversee the clinical development of Tibsovo at Servier, ranging from a broad range of different malignancies in the hematology as well as solid tumor space.
Thank you. Patrice, would you mind going next?
Thank you, Robert, and good afternoon, folks, and thank you for inviting me today, and good to see Susan and Josh here. My name is Patrice Lee. I'm VP of Pharm Tox and Exploratory Development at Pfizer, the Boulder site. And I am the non-clinical research lead for the Braftovi and the Mektovi franchise at Pfizer. Braftovi and Mektovi, of course, are BRAF and MEK inhibitors respectively, and they were approved in 2018 for the treatment of BRAF mutant metastatic melanoma. And we're very happy to say that 2 weeks ago, we got another approval for the doublet in BRAF mutant lung cancer. So truly trying to expand the use of this doublet to treat as many patients as possible.
Fantastic. Thanks, Patrice. And Josh, would you mind taking us home with your introduction?
Fantastic. Thanks so much, Robert. And I'm honored to be on this panel with Susan and Patrice. So I'm the global medical head for Rybrevant development at Johnson & Johnson. And I'm just thrilled to be here. I'm actually calling in from Madrid, where we just presented a lot of exciting data at ESMO. So I'm just so excited to be here to talk with you all.
Fantastic. Thank you all. One brief reminder to the audience today. You should have access to a link to share a statement. I'm sorry, you should see a statement that has been submitted, not the other way around. By all means, take a look at that information and review it as we're going through today. So I'd like to start the discussion today, folks, about the products that you're each here to represent. So let's start off a little bit broad. Why did your company choose to focus in the disease state and indication that you're here representing today when there are a lot of different unmet medical needs? So maybe you wouldn't mind sharing some of that. Patrice, if you wouldn't mind kicking us off, we'll round robin around the crew.
Sure. As you know, Pfizer acquired Array BioPharma in Boulder back in 2019, and Array BioPharma was well known for being successful in discovering and developing tyrosine kinase inhibitors for various different targeted cancer targets. So we invented Mektovi, and then we knew that we needed to partner with another company in order to bring out the best of the MEK inhibitor, and that was when we partnered with Novartis. And we knew that there was still an unmet need in this space, despite there being 2 doublets before us, because although we were not first in class, our goal was to be best in class, because there was an unmet need in the space. And so we moved it forward initially with melanoma, And then more data came out showing that, BRAF mutations are involved in a host of other cancers. And perhaps we could even expand into that. And as I mentioned, we just got the lung cancer approval, but there are plenty of other cancers that utilize mutations in this BRAF pathway to escape other types of treatment.
Fantastic. Josh, would you mind also sharing your perspective on why the company chose to focus in the area that you have?
Yeah, thanks so much. Johnson & Johnson's vision is to cure cancer. So we're committed to pursuing transformational therapies, finding cures to intercept cancer in patients that are of high unmet need. Rybrevant, or amivantamab, it's a bispecific antibody targeting EGFR and MET, and it's actually the first bispecific antibody that has ever been approved in solid tumors. And it's also the first targeted therapy to be approved for patients with non-small cell lung cancer harboring an EGFR exon 20 insertion. Lung cancer is one of the most common types of cancer. It's the most common cause of death. And while EGFR mutations are the most common target, most these patients with exon 20 insertion do not respond generally to the approved targeted agents for more common EGFR mutations. And so we saw that amivantamab or Rybrevant had a meaningful activity response rate, about 30%, median progression-free survival, about 4 months. And so one of the reasons we think this is that Rybrevant targets the extracellular part of both the EGFR and MET receptor, which allows you to bypass the intracellular issues with the pocket where the TKI cannot bind as well. It's deliberately designed to have differing affinities for EGFR and MET, and it also can recruit immune cells to potentially lead to more prolonged responses. So we're very excited about this molecule and the impact it can have for patients.
Fantastic. Thanks, Josh. And Susan, would you mind also sharing your perspective, please?
Yeah, absolutely. IDH mutations have, I would say it's probably one of the newer oncogenic targets when we think about, different actionable oncogenes throughout our understanding of biology of cancer and its development across a whole variety of different solid tumor and heme malignancies. So IDH1 mutations were first uncovered as potential actionable targets in both hematologic malignancies and solid tumors in the early 2000s. And at that time, I was working at a smaller biotech company who was developing the first-in-class and IDH1 mutant selective inhibitor, which was Tibsovo. So this is an oral targeted therapy that targets the IDH1 mutation, and it's shown efficacy across both hematologic as well as solid tumor indications. Servier acquired the oncology business of Agios, as well as several individuals who were working there chose to join Servier in April 2021. So gratefully, I've been able to continue with this program and advance the development of this novel first-in-class therapy for patients in a variety of different settings.
That's fantastic. Thank you. And thank you all for sharing that information about your organizations and the products. I'd like to talk a little bit about the clinician response to the products. Was there a measurable impact that you could cite from the clinical outcomes for patients? Maybe, Josh, if you don't mind kicking us off, it sounds like there may be some rather interesting things that you have to share from just recently as well.
Yeah, I mean, this is really hot off the presses. We were very excited. So Rybrevant was first granted breakthrough designation 2020, and then approval by the US FDA May 2021 in exon 20 after progression on platinum-based doublet. But at ESMO this past week, we were able to present 3 phase 3 clinical trials, all presented at presidential symposia here at ESMO, that showed that Rybrevant led to markedly improved outcomes. So the first one was in Papillon, which is a study evaluating chemotherapy with Rybrevant versus chemotherapy alone. This study showed that amivantamab and chemotherapy led to a 60% reduction in the risk of progression or death, consistent benefit across all subgroups. Mariposa 2, another study evaluating amivantamab or Rybrevant with chemotherapy, this time in common EGFR mutations, found that there was a 52% reduction in the risk of progression or death for these patients after progression on osimertinib or Tagrisso. And then most Most recently, we saw Mariposa, which was our first-line study in the largest patient population. We had 1,000 patients in this study, and we were able to show that even up against osimertinib, Tagrisso, TKI, standard of care in this population, there was a 30% reduction in the risk of progression or death with the combination of Rybrevant or amivantamab with lazertinib, third-generation EGFR TKI. And in all three of these studies, there was an early interim trend towards an improvement in overall survival. So we're just really thrilled that this molecule seems to be impacting patients across the wide range of EGFR mutations, and we're just really excited to try to bring this to patients.
That's fantastic. That's great news to share, Josh. Thank you. Susan, I'll turn to you next to tell us a little bit more about the IDH space.
Yeah, so it's interesting. IDH1 mutations actually occur in a variety of disease settings where there was very little progress. So we initiated our first in-human studies in 2015 and 2016 across both heme and solid tumors. And at that time, very little progress had been made in acute myelogenous leukemia, which is a hematologic malignancy that's acutely life-threatening and requires emergent intervention. Tibsovo showed very promising complete remission rates as well as transfusion independence, which is really an important clinical benefit parameter for patients suffering with this disease. Tibsovo was first approved by the FDA in 2018 and then a subsequent approval in 2019 in a first-line indication for patients with AML harboring an IDH1 mutation who are ineligible to receive intensive chemotherapy. So we took our understandings and learnings from the hematology space with respect to the biology and what we were learning about this very novel mechanism of action of Tibsovo, and we applied it in the solid tumor setting, which was really very interesting because IDH1 mutations that occur in solid tumors happen to occur in disease states where either nothing had ever been approved or treatment paradigms were really an extrapolation from other types of malignancies that are more frequently seen in the clinic. Cholangiocarcinoma represents one of those disease entities. It's a rare biliary tract cancer, and IDH mutations occur in roughly 15% of patients with the disease. So it's a rare disease within a rare disease setting. And at the time when we were initiating our phase 1 program, followed by our phase 3 Clarity trial in 2017, there were no approved therapies for patients with cholangiocarcinoma. So unlike the progress that we've seen across a variety of other disease settings, unfortunately for patients suffering with this disease, they were relegated to receiving fairly antiquated chemotherapy regimens. So Tibsovo really represented a novel therapeutic option for patients with an IDH1 mutation, and it also represented the first targeted therapy approach for this patient population. When we conducted our phase 3 clinical trial, the primary endpoint was progression-free survival. The study recruited in about a 2-year period of time, which is remarkable for a rare cancer setting, and hit the primary endpoint of progression-free survival based on blinded central review. And what that amounts to is a 63% reduction in the risk of progression or death among patients who had previously received chemotherapy. So again, this is an aggressive cancer. Once patients have progressed, unfortunately, following chemotherapy, their disease can take a very aggressive course. And to have a 63% reduction in progression or death is really a clinically meaningful outcome. Other clinical benefit parameters, I think, are really important to note, and those include a safety profile that was tolerable for patients, as well as an overall trend in overall survival. And then subsequently, our health-related quality of life parameters also supported clinical benefit with respect to preservation of quality of life, which is also essential, where the aim of treatment at this stage is really to prolong patients' survival as well as their interval from progression, but also maintain quality of life when you can't expect a cure in this disease setting where the patient's disease is already advanced or metastatic. So after we received FDA approval in 2021, what we've learned is that, practitioners now consider Tibsovo to be a standard of care option for patients with IDH1 mutated cholangiocarcinoma who've received previous lines of chemotherapy. It's also listed as a category 1 recommendation in the current treatment guidelines, again, indicating its clinical value for patients with this disease. And then finally, through our patient offset survey, we've maintained a very close connection with the patient advocacy organizations, which is really a critical step towards making innovative progress for patients, especially when you're in the rare disease cancer space. And we've heard very powerful testimonials from patients that gives us a glimpse into the experience they're having on Tibsovo.
Susan, that's fantastic. And I'd love to come back to this concept of patient advocacy, patient involvement towards the end of our discussion today, because I think what you've just shared is very powerful. So, Patrice, I'd love to hear your perspective as well around the impact and clinical outcomes for patients associated with Braftovi and Mektovi, please.
Thank you. So as you know, the approval of Braftovi-Mektovi in 2018 was based on the Columbus trial, and this was a phase 3 global randomized 1-to-1-to-1 active control open label multi-center trial that involved 577 adult patients with BRAF mutant V600E and K mutant unresectable metastatic melanoma. So patients received Braftovi plus Mektovi, Braftovi alone, or vemurafenib as that positive control. And the major efficacy outcome was PFS as assessed by BICR. And the post hoc analysis for the PFS, which is the major efficacy outcome, showed that with Braftovi plus Mektovi, PFS was 14.9 months compared with 7.3 months in the control arm with vemurafenib alone. So that was very impressive. And I think this is having an impact on, the physicians prescribing BRAF and Braftovi and Mektovi right now. We have over 15,000 prescriptions for the combination in this later line setting of melanoma. Now for the approval in lung cancer, that was actually a phase 2 study, and it was an open label trial where we showed remarkable benefit of the combination of Braftovi and Mektovi in patients with BRAF mutant non-small cell lung cancer. And the major efficacy outcome was ORR per RECIST 1.1 and by BICR. And we had a 75% ORR in treatment naive patients, as well as a 46% ORR in previously treated patients. In the prior group, the treatment naive patients, we had a 15% complete response rate, a 59% partial response rate, and then in the previously treated patients, complete response rate was 10% and partial response was 36%. And thus the FDA approved this doublet on this phase 2 open label study in about approximately 100 patients with BRAF mutant non-small cell lung cancer.
Outstanding. Patrice, thank you so much for sharing that. I have one question to sort of wrap up this more structured component, and then we'll open it up to more of a discussion among our panelists. So, Susan, we'll start with you on this one. Can you talk a little bit about some of the challenges that you had to confront when you were transitioning this from this early proof of concept into human trials, and then ultimately getting approval and market authorization and everything like that? Are there any lessons that you feel comfortable sharing from your experience that might help some of the folks on the line get a better feel?
Yeah, and I touched on it a little bit earlier about in the rare cancer space, it's really important to learn from patients, patient advocates, and also work very closely with the different investigators and sites and the thought leaders in the space. As a former practicing oncologist, I've seen both sides of the spectrum, having been at the bedside treating patients and having a paucity of options to offer them, especially when it comes to clinical trials. So for me, as a clinician working in an industry space, it was really, really important to get feedback from patients on the design of our study, importantly, our phase 3 trial. When we were working on our phase 1 efforts, we certainly were already engaged with patient advocates to at least learn about the patient journey, learn about the symptoms associated with the disease, and also how could we best tailor our phase 3 program to meet their needs, but also meet the regulatory hurdles that are absolutely required for an approval path. So one of the steps that we took was to actually present our study design to a very noteworthy patient organization here in the United States, as well as one based out of the UK, to really get the feedback on our study. And what was important to note is that in cholangiocarcinoma, there was no standard of care. So the design that we had originally opted for included a placebo control. And with that in mind, you can imagine patients had some fears about being randomized to a placebo control, and we certainly received important feedback from them, which we did incorporate ultimately into the final design. And that essentially meant allowing for crossover, which is a design feature that you oftentimes will see in placebo-controlled trials, and in some cases you may not see it. But we listened to the patients. It was really important for us to, again, have a study design that would meet their needs, but also provide clinically meaningful outcomes so that we would then have an approval path. Crossover effectively means that patients randomized to the placebo arm at the time of radiographic progression would have the opportunity to then cross over and receive active therapy. So while this complicates an endpoint known as overall survival, it was still something that I, in particular, as well as the team and the company, felt very strongly about incorporating, again, listening to the patients and meeting their needs. I would say that to me is a very important step to take as a drug developer in pretty much any indication, but especially in the rare cancer space where you absolutely need to be working very closely with the advocates in the community in order to recruit the studies and raise awareness around IDH testing in particular. The other piece of what I would say is the learnings, and I think many of us have lived through this over the past few years, is how do we modify clinical trials operationally in such a way to ease the burden on patients? The COVID pandemic really forced us into a situation by which we had to be flexible. We had to be very thoughtful in what we were asking of patients and sites to be doing. And through that effort, I think we've realized that, maybe not everything requires an in-person visit. Maybe we don't need as many blood draws, et cetera. So taking those learnings and moving them forward for patients, I think is really critical. And it can only help with recruitment efforts. And again, help us in increasing the diversity in our clinical trials. So once again, we can learn as much as we possibly can through the efforts that we put forth.
Awesome. Thank you, Susan. Patrice, would you mind sharing some of your own challenges that you've helped to overcome in this process as well?
Sure, thank you. And I'm speaking again from the non-clinical perspective as well, since I was primarily in that realm, not from the clinical perspective. But I think it was really important early on when we were Array and a small biotech that we could recognize the unmet need that was still out there in this particular space and try to be best in class with any therapy we brought forward. And then we were lucky to have Pfizer acquire us, so that we now had the resources to actually prosecute this program and allow it to move faster, because that's the most important thing, getting it to patients faster. And I echo a lot of what Susan said about the challenges that have come up recently with COVID and being able to enroll even our current studies with the doublet and make the burden lesser on patients, because that is really important.
That's fantastic. Thank you. And Josh, same question as the other two panelists, please.
Yeah, I mean, I think that we had challenges first on a molecular basis. So, amivantamab, as I mentioned, is a bispecific antibody. So first, we know that EGFR is highly expressed and MET is more expressed on tumors. So one of the things that we chose to do was make the MET arm have a higher affinity than the EGFR arm to try to optimize the toxicity profile. Because we view amivantamab as precision medicine, trying to precisely deliver EGFR inhibition at the site of the high MET expression. Now, obviously, as Susan and Patrice have both mentioned, COVID had a huge impact on our study. EGFR mutant non-small cell lung cancer most commonly occurs in Asian patients. And so we had a lot of patients enrolled in China as well as Asia. As COVID hit, we were opening our trials and executing these studies. So we had to think on our feet, and there was working with the sites to have remote monitoring, direct shipment of agents to patients as needed and as appropriate. But one other point that I'd like to highlight is throughout our trials, we also were dealing with the conflict occurring in Ukraine. And so we had sites that were open in Ukraine. And this was honestly one of the most moving aspects. So like Susan, I'm trained as a medical oncologist. I've joined industry relatively recently. And it was enormously moving to hear my team say, okay, we have this many patients that are in Ukraine. How do we get our medicine to them in a war-torn country? How do we bring our patients to the site, to a safe site, so they can be safely treated? And we had patients that moved from one site to another. We were really very nimble in terms of how we handled this. And I'm very honored to say we didn't lose any patients to follow-up in that process. And it was truly a remarkable team effort that led to that success. And it was a very difficult time, certainly for our patients, most of all. But in terms of us supporting those patients, it was a real mission for the team to help them.
That's fantastic. And it actually speaks to, I think, what would be a good topic of discussion amongst the three of you, if you don't mind. And this is more around this concept of the patient advocacy, improving that quality of life. And I guess really it's what advice would you give to some of the folks listening if they were trying to emulate some of the things that you've done successfully, right? If they want to move from success to success successfully, how do they do that, right? And in this regard, and Patrice, I don't know if this would be something that you would feel comfortable maybe sharing your thoughts on first, but let's keep this as a discussion for this portion, if you guys would like.
Yeah, I'm not as qualified as Josh or Susan to discuss this, but one thing I noticed just from observation and being in this business for about 35 years is that you need to work collaboratively with the regulatory authorities. We have had great results working with the FDA early and often to make sure that we could do things more nimbly and reach the patients more quickly and get our trials underway. And I would love just to sit back and hear Susan and Josh discuss their personal stories on this.
Okay. Yeah, please.
Yeah, no, I'm happy to go first. Oh gosh, what a great question. It's one I feel really, really passionate about when I talk to my teams, and even just making the transition from academia to industry is just this idea of the sense of urgency, right? Never let that leave, your realm of, vision and what is guiding you. And I think that, that really stems from the unmet need and really humanizing what we're trying to do. And not everybody in industry roles have had the benefit of either working with patients or hearing from patients, or may have even, experienced going through, the treatment course of, a patient or a loved one with cancer. So I think as much as you can bring that to life within a company environment, it can really be immeasurably impactful for the entirety of the organization. And I think it really takes champions within an organization to rise to the occasion, be willing to take calculated risks. Even if you don't have all the information in front of you, you may have enough to make the right decision. And what I would say is just sort of the hallmark of the work that we've done here at Servian and my former company is to involve patients early, really understand what it is that they need, understand the biology, understand exactly what you're trying to achieve in your first in human study so that you can get the most out of those efforts to help guide decision-making for late-line registration enabling efforts, or perhaps even, as was the case for us, use your phase 1 data as a registration enabling trial. So we have to be creative, and that sense of urgency, I think, is what guides our ability to be creative. I think having a mission-oriented approach as well, as many of you have already commented on, a genuine concern for the patients, number one, but also what you're trying to achieve with the powerful science that you've harnessed. And try not to leave too many loose ends, try and go big as much as you can and be ready to fail. And I think that's perfectly reasonable. That's what we have to do in order to innovate. And if we don't take risks, we're never going to make the progress that patients are expecting of us.
I love that. And candidly, as someone in the tech side of the healthcare ecosystem, we do have a very move fast and break things, as everyone knows, in Silicon Valley mindset, right? And I think that allows that innovation to happen, right? When you're not constrained by fear of failure, right? You can actually reach pretty impressive successes. But Josh, I'm curious to hear your perspective on the patient involvement and all of that as well.
Yeah, I spent 10 years as a lung cancer doctor. And lung cancer has been, lung cancer is the leading cause of cancer death. That's a huge issue. And yet there has been this molecular revolution in lung cancer, identifying all of these individual targets. And what is truly inspirational is that the people who are driving that success and the people who are driving that research, it's often the patient advocacy groups, and working with them when I was in academia, I saw that passion. I saw that drive, because you don't have to question where the sense of urgency is coming from. This is their lives, their loved ones' lives that are literally at stake. And so what I'm so thrilled about in working on Rybrevant is that, we can identify in exon 20, there was no target. This is something that, just like the other EGFR mutations, they identify this. This is usually never in light smokers, younger persons, women more frequently. There's you have a molecular driver. We know exactly how your tumor works, but we have no targeted therapy to help you. And so this is something where, with the approval of Rybrevant in the second line, that was the first hint of how we can help these patients. But being able to bring the Papillon data, where bringing that targeted therapy up front, just like every targeted therapy, this is just the first step on a long road to try to help these patients. And it's just absolutely inspirational to speak to these patients, to hear their stories. And just at the end of the day, if the patients are the center of your mission, you are always going to make the right decision.
I couldn't agree more, right? I think oftentimes this idea of rare versus common types of cancer, right? I think the reality is it's always personal. And that means that you always will have that degree of fear, as Susan, you spoke to in some cases, right? Where folks are a little bit hesitant or struggling in that regard. So I appreciate you guys sharing this. This is super helpful. I do have one other question, and I do want to remind the audience, please feel free to submit questions as we can. I'm going to be taking a look at that, but I'd like to ask you guys a little bit about what you think is coming, right? So as scientists, as clinicians, what do you see as like this next wave of innovation, right? Specifically for your therapeutic area, right? But what other innovations are going on in the space that might be worth considering in this field in 5 years? Josh, do you mind kicking us off?
Yeah, sure. Absolutely. So, from my perspective, we have focused all of Rybrevant's development in lung cancer. And I'm a lung cancer doctor. That fits right into my wheelhouse. I get that. But what we also know is that, at least for our molecule, EGFR and MET are driving alterations in a wide variety of malignancies. So we are focused at Johnson & Johnson on exploring how we can utilize Rybrevant in those cancers, in colorectal cancer, in head and neck cancer. How can we bring improved outcomes to those settings? And then just more globally, the advent of artificial intelligence, utilizing these sorts of technologies to identify new targets, build new drugs, and help more patients.
Thanks, Josh. That's fantastic. Patrice, would you mind also sharing your perspective on this and innovation in the next few years?
Sure. And I agree with Josh regarding looking beyond your particular tumor type. And as I mentioned, Braftovi, Mektovi are approved in 2 different tumor types now. Braftovi in combination with cetuximab is improved in colorectal cancer. So the tumor agnostic approach, I think, is where we're going to have the most bang for the buck here, and finding these common driver mutations across the different tumor types. I think AI can help with that. I don't want AI to give AI the ball. I want to use it as a tool, because I still believe in spontaneity and curiosity of the humans, and also the responsibility we have. But I look forward to that as well going forward.
Patrice and Susan, I promise I will jump to you in a second, but you've both mentioned AI at this point. I am curious, Patrice, it sounds like you have a little hesitance around it, and understandably so. Would you mind sharing some of those dimensions? I think the audience may be interested in your perspective on this.
Yeah, this is just my personal perspective. I'm not speaking for Pfizer, but I think there's a place for it. And as Josh mentioned, I think it'll be ideal to start off by using it to pick the right targets. So many people go down this long road working on a program to only realize, well, it wasn't what we thought it was. So when you have complex, large data sets, I think AI would be great to help you narrow down your focus so that you don't spend a lot of time and money and going after the wrong target for a patient who actually needs a drug for the right target. But the only reservation I have is that I I'm not, invoking the Terminator here, but there comes a point
I'll be back.
I knew you were going to say that. Where, I mean, it's only as good as the information fed into it initially. But I just look forward to learning more about it and seeing how it can help us, especially on the non-clinical discovery side, because I mean, some folks have said, well, we've run out of targets.
Yeah.
I don't believe that. I just don't think we know where they are yet.
Absolutely. And thank you for that. Would anyone else care to comment on this topic before we pass the mic to Susan to hear what's coming in the IDH space? Okie doke. Susan, feel free, please.
Yeah, no. So for us, so with Tibsovo, the original approvals in 2018, 2019 in AML, these were monotherapy indications. And then in 2022, in combination with standard of care chemotherapy, azacitidine, in patients who had IC-ineligible AML. So these were all, major successes in the hematology space. And then specifically for cholangiocarcinoma, our monotherapy approval in 2021. But we wanted to take those approvals and the benefit that, patients were receiving more globally. So I think one of the major initiatives for us was to be able to submit our data worldwide. And as of, this past year in 2023, we've been able to extend the benefit of Tibsovo to European patients with EU approval in both cholangiocarcinoma as well as in AML with azacitidine. So it's really great progress because, again, our mission is to help as many patients as we can around the world. Tibsovo is a first-in-class IDH inhibitor, and it's the only approved IDH inhibitor in the EU now. So, hopefully we're going to continue to expand beyond into the rest of world regions. We also have recently submitted our relapse refractory myelodysplastic syndrome data to FDA. It's currently under priority review, and this also has breakthrough therapy designation. So we're excited about extending further into the hematologic space. Myelodysplastic syndrome is a precursor disease to AML, and about 30% of patients, unfortunately, their disease will progress to AML. So this represents an important opportunity for this patient population, hopefully, again, to provide clinical benefit to them and reduce the risk of AML transformation. What I would say I'm really, really excited about in the IDH inhibitor space is our program in brain tumors. So our sister product, vorasidenib, is a dual mutant inhibitor of both the IDH1 as well as IDH2 enzymes, and it's specifically designed to be brain penetrant. So similar to Tibsovo, it's an oral therapy. And as many of you probably know, IDH mutations actually occur at a very high rate in patients with low-grade gliomas, up to about 80% of patients. So it represents a very relevant target for this patient population, who typically are quite young. Median age of diagnosis is around 40. And over the past 2 decades, we haven't really seen major progress in this space. At this past year's ASCO, we were able to present our phase 3 Indigo study at the plenary session. It was the first presentation at ASCO. So it represents a potential major breakthrough for patients who have IDH mutant brain tumors, in particular gliomas. And I think this would be a major advancement for the field at large, one that's been long awaited. So I'm very excited about that. As far as additional work that we're doing, we're very interested in novel combinations. So beyond standard of care combinations, as is the case in AML, we are very interested in understanding the impact of IDH mutations extracellularly. We've learned that the oncometabolite 2-hydroxyglutarate actually has an impact on the tumor immune microenvironment. So now we're venturing into novel immuno-oncology combination approaches across our disease settings. So I think there's a very bright future ahead for us, and we're really looking forward to advancing further for all sorts of disease indications beyond what we've already studied.
Fantastic. So we actually have a question in the Q&A. So quick reminder, everyone who is holding on to those questions, now's the time. Put them in the Q&A. It's down at the bottom of your screen. You should see it next to the share screen, chat, record, all that. You can pop them in there. So Christopher Henschel has asked, are there any disadvantages in using therapeutic bispecific antibodies in oncology?
So I think this one might be for me because I'm developing a bispecific antibody in oncology. So So I'll take it. But if others have comments about it, I'm certainly welcome to that. Look, I mean, so a bispecific antibody introduces a lot of positives, right? You can target different types of tumors with differential markers. You can get more. And with immune cell redirection bispecifics, you can also bring immune cells to the mix. And that can introduce differential toxicity because you're bringing in immune cells, cytokine release syndrome, things like that. For us, what it does is you have two safety profiles that you have to focus on. So we have EGFR related toxicity and we have MET related toxicity. And so that is an element that you have to consider when combining with other agents, when developing your clinical trial and your adverse events of special interest. But in my view, the benefits far outweigh the risk, at least if the bispecific has meaningful targets and can get patients to good response levels.
Thanks, Josh. If there are any more audience questions, please feel free to keep them coming in. We have about 10-ish minutes left. I will actually break out the soapbox and ask each of our panelists if you would like to step up on it, by all means. I will give, I think it would be beneficial 2 minutes if there's anything that we want to say to this audience. Soapbox is yours. Does anyone want to start? Anything you would like this audience to be aware of, keep in their heads. If not, totally fine.
Yeah, as I mentioned earlier, there's always this argument, at least in the non-clinical and late discovery world, about best-in-class versus first-in-class. And I mean, my bias from having both brought forward first-in-class, best-in-class, and then first-in-class, that don't give up if you're not the first one there, because what we know, cancers learn now, and they're escaping through resistance mechanisms. So don't just give up because you think that the cancer type has met the need from whatever pharmaceutical is being brought forward. Because eventually something's going to come up like resistance where you'll need to go back in and address it again. So try all of them. Be best in class, first in class, and both.
That's fantastic, Patrice. Fantastic. Josh, I don't know if there's anything that you or Susan would also like to add.
Yeah, I mean, I think that one thing that I would recommend is to make sure that you're staying true to the science. And just because everyone else is in a certain class of drugs, or you're seeing lots of those coming down, or everyone's focused on a certain type of agent or certain area, a certain pathway, that doesn't mean that you need to as well. Go based upon what the science is telling you and what your training has shown you, and listen to your team so that you can get the best results. So as I said before, we all have the same goal. We all want to make cancer a thing of the past. And the only way that we can do that is by using team science and bring the best technology to the fore.
Fantastic. Susan, anything to add?
No, I would just probably build off of both of those statements. I agree. I mean, I think it's important not to be risk averse. I think it's important to, yes, be faithful to the science, but also be willing to question, the science as well and extend beyond your own internal understanding. And that means forging collaborations globally. Within our backyard here in Boston, we have so many great incubators and people that are really at the cutting edge of technology, and really try and take the time to learn those opportunities and take the time to meet with people so that you can give your program the best shot towards success. And then I've said this, I think multiple times throughout the panel, but, involve yourself with patient advocacy organizations, really understand the disease areas from the patient perspective. I think this to me is going to be absolutely instrumental in order for us to make progress, especially in these rare biomarker-defined subset of cancers. And I absolutely agree with the rarity statement, but I think there's a feasibility as well and challenge that some companies are fearful of. They will say, oh, it's so small, So for commercial purposes, maybe there's a reason they don't want to do it, but also feasibility purposes. People get very nervous that, oh, how could we enroll this? But the reality is you can, and there are several examples. And I think, the program that, I've presented to you represents one of those. And I think there are countless examples. So don't shy away from that. Let the science lead you and let the patients lead you as well.
That's fantastic. This has been such great insights, everyone. I don't see any other questions coming in, so I think we are happy to wrap. Now, any final comments from our panelists regarding our time together today, or just anything else that you'd like to share?
Thanks so much for having us. It's been a real pleasure to be on the virtual stage with you, Susan and Patrice, and great talking with you as well, Robert.
Yeah, I would say the same. Thank you so much. I love learning about your programs. I'd love to meet like-minded pioneers, and I hope to meet you all in person sometime soon.
I concur. Maybe we'll all be in New York City, or Susan, you're not going to make it this time.
I'm going to make it this time. I will be there this time.
I'm just coming back from Madrid. I can't make it this time.
Yeah.
Well, in the future, when you guys do get together, it'll be a hell of a brain trust. So I want to thank you all. Obviously, Susan, Patrice, Josh, thank you for joining us all. For the folks in the audience or folks watching along at home, this is going to be available on the Galien Foundation homepage. So by all means, check it out. Thursday is the forum. It's going to begin at the Alexandria Center for Life Sciences at 7:30 a.m. And feature keynote of pharma CEOs from the US and Europe, concluding with a presentation of the 2023 Roy Viglios Pro Bono Humanum Award winner, followed by the awards gala, both of which will be held at the American Museum of Natural History. So we hope to see you there. Thanks again, folks, for the time together today.
Thank you all. Bye-bye.
Bye.
Bye.